Good morning, afternoon, evening, whenever you end up watching this. Um, this is our first Zoom PowerPoint lecture coming at you from my house because, social distancing, y'all. Um, yeah, this is my husband's art studio with all the books behind me. But I'm just going to cover the rest of chapter 22 real quickly for y'all. And then by now you will have already seen all the announcements, hopefully, all the announcements, all the kind of details of how this is going to work. My plan is to not have hour-long lectures for y'all. I'm going to try to break it up into little snippets and really only cover slides that I think are super important, maybe harder to understand. And then we'll figure it out from there. Of course, the PowerPoints are online already for y'all. I'm sharing my screen here so that we can kind of see both at the same time. I have a completely Jimmy rigged thing going on here, but it's going to work. So if you have any questions at all, please see the discussion boards that I've started. I'll have a discussion board for at least every chapter of material, if not for every lecture that I do. I think probably for every chapter. I don't know, we'll play that by ear. Ask questions there. You can always message me privately, email, whatever, okay? I'm as available to you as I possibly can be, really even more so now because of all of this. So let me just finish up chapter 22, and I got some other lectures to record, so we'll get started. Okay, so the last thing that we were talking about, man, seems like a lifetime ago, was the matching of ventilation and perfusion. And so we know that our lungs' job is to oxygenate our blood. And if we have less blood flow to a certain part of our lung, it makes no sense to send a lot of air to that part of the lung. Less blood, we want to match that with less air. More blood, more air, because the purpose is to oxygenate that blood and get the carbon dioxide out. And so we talked about how our body will match. Let me change this real quick. Annotate. I'm going to use a little spotlight tool. I don't know if y'all can see that. Okay. We match the ventilation with the perfusion. If ventilation is less than perfusion, then we're going to vasoconstrict those blood vessels down and send less blood there. If it's more than perfusion, we have more air into that area than we have blood flow, then we're going to vasodilate and bring more blood to that area. All right. No. Okay. Go away. All right, we'll do it that way. Okay. Still figuring all this out. All right. So internal respiration, remember, is the process by which we are sharing oxygen or exchanging oxygen and carbon dioxide within our tissues or at our tissue level, from capillary to tissues. This is all driven, once again, by pressure gradients, specifically the partial pressure gradient of oxygen and carbon dioxide. And when blood's going into the tissues, the oxygen level in the tissue is always going to be lower than the oxygen level inside the artery. And so it is going to push oxygen from the blood to the tissue, and opposite for carbon dioxide. There's more carbon dioxide in the tissue, less in the blood vessel, and so it's going to push carbon dioxide there. So venous blood has a partial pressure of oxygen of 40 millimeters of mercury by the time it leaves the capillary bed and goes off, and a partial pressure of carbon dioxide of 45. And this is a similar graph to what I showed y'all earlier. I'm going to put this annotate back on, sorry. That I showed y'all earlier here, but we're down here at this point. We're at the blood leaving the tissues. Partial pressure of oxygen is at 40, carbon dioxide is at 45. Whereas before, the blood that was coming in, the partial pressure of oxygen was at 100 millimeters of mercury, and carbon dioxide was down to 40. There we go. Okay. So how these gases are transported in our bloodstream. Let's talk about that for a little bit. Oxygen transport and the carbon dioxide transport. Of course, those are the two that we're worried about. For oxygen transport, there is a little bit of oxygen that is carried just within our plasma. Very little, about 1.5% of it. Most of our oxygen is, as we've talked about before, carried in our or in our red blood cells attached to the iron portion of our hemoglobin. Remember that there's 4 hemoglobins. No, I'm sorry. There's 4 hemes for every hemoglobin, because there's 4 globin molecules that each have a heme inside the middle of it. And so you're going to have 4 oxygen molecules per hemoglobin molecule. Now, oxygen and hemoglobin, when they combine, we sometimes call that oxyhemoglobin. And when they come apart from each other, when oxygen leaves hemoglobin, which it does because we need it to, it's called reduced hemoglobin or deoxyhemoglobin. And so there's this backwards or to and from reaction with hemoglobin and oxygen. They bind together, they break apart, they bind together, they break apart, whatever the demands of our body is or where that blood is. So when oxygen binds to hemoglobin, hemoglobin actually changes its shape a little bit. As oxygen binds, hemoglobin becomes more readily bound to oxygen. Its affinity increases. When you have one oxygen binds, it's more readily going to bind another oxygen and yet another and another. It's not a linear curve. In fact, I'll show you a curve here in just a second, but as more oxygen binds, the more the affinity of hemoglobin to oxygen is. The opposite is also true. As oxygen leaves the hemoglobin, hemoglobin becomes has less affinity to oxygen, and so it dissociates more readily. Now, fully saturated hemoglobin, again, 100% saturation, has all 4 heme groups are saturated. They have 4 oxygen groups on there. Anything less than that, we call it partially saturated. So even if just 3 of the 4 are saturated, it's a partially saturated hemoglobin. Right, and so how this rate is affected is by the levels of oxygen that are in our tissues at any given time. So there's a lot of factors that are going to be at play here. The partial pressure of oxygen is one, the temperature, blood pH, the partial pressure of carbon dioxide, and even the concentration of a chemical called biphosphoglyseric acid, or BPG. This is something that's built up when we have low oxygen levels. If we're turning to an anaerobic way of making ATP, we'll talk about that after this next exam. We're not using oxygen. We're building up a little bit of lactic acid, and we're also building up this BPG. Right, so the effect of the partial pressure of oxygen on hemoglobin saturation, that when we're looking at this oxygen hemoglobin desaturation curve, it is not a linear curve, as I mentioned before. It's an S-shaped curve because as one oxygen binds, it has more affinity for more oxygen. So it's not linear. It's this weird kind of S-shaped curve thing. So you can see that here. If you're looking at this graph, this left, the y-axis, wow, forgot my algebra there for a second, is the amount of oxygen that is bound. So, you have 25%, 50%, 75%, 100%, right? Depending on how many are there, or 0%. This other axis is the partial pressure of oxygen. So, arterial blood is up here at 100 millimeters of mercury. Down, in our venous blood, it's at 40%, or I'm sorry, 40 millimeters of mercury, right? And so let's say we have one oxygen that's bound to the hemoglobin. Well, then it's has even more affinity toward the next oxygen, and the next oxygen, and the next oxygen. So you can kind of see this S-shaped curve. In the lungs, where the partial pressure of oxygen is high, we got a fully saturated hemoglobin up here. Down here in our tissues or in our venous blood, we're at about 75% saturated. All right, we'll talk about why we're only using 25% here in just a little bit. All right, so in arterial blood, again, the partial pressure of oxygen is about 100 millimeters of mercury. That contains about 20 mils of oxygen per 100 mils of blood, 20% volume. And our hemoglobin is pretty much 100% saturated, 98 to 100%, somewhere around there. If you were going to go and take your pulse ox, you're wanting to see upper 90s for somebody who's having, especially if they're having some kind of lung problem, coronavirus per se. You're looking at upper 90s. That's really where you want to be. So any further increases in the partial pressure of oxygen really doesn't do us much good. Let's say we take the partial pressure of oxygen is at 120 millimeters of mercury. Well, we're already basically at 100%. So that's not going to do us a whole lot of good, right? In venous blood, on the other hand, the partial pressure of oxygen drops down to 40 millimeters of mercury. That's about 15 mils of oxygen per 100 mils of blood. And our hemoglobin is 75% saturated. So that 75% that we still have left, we call it the venous reserve. And this is a really good thing to have because, let's say, in the case of this graph right here, at sea level, which is kind of where, at least I don't know where y'all are right now, where Central Texas is, we're at about 100 millimeters of mercury for our partial pressure of oxygen in the atmospheric air. And so we're hanging out 98 to 100% fully saturated there. But if we go up to Colorado, where there's less oxygen in the altitude, let's say it drops down to 80 millimeters of mercury right here, then you can see that our hemoglobin is still 95% saturated at that point. And so that's a good thing. We can go up to really high altitudes before seeing really desperate drops in the saturation of our hemoglobin, right? Now, of course, there's other heights like Mount Everest where, yes, you have to have oxygen because there's just simply not enough there, or if you're in an airplane or something like that. But normal circumstances where people normally are, that's not a problem because of this venous reserve that we have. All right, other factors that influence hemoglobin saturated: increases in temperature, increases in acid, increases in the partial pressure of carbon dioxide and that little chemical PPG. These all modify the structure of hemoglobin. They decrease its affinity for oxygen. And this occurs more in the systemic capillaries, and it helps to enhance oxygen unloading from our tissues because it's making oxygen and hemoglobin have less affinity for each other. So it's a good thing. It's encouraging oxygen delivery to our tissues. And it also shifts that S-shaped dissociation curve to the right, which means oxygen and hemoglobin have less affinity for each other. When you shift that curve to the right, they have less affinity. When you shift it to the left, they're more attracted to each other. Okay, it's important to remember that. If you decrease any of these factors, less acid, colder temperatures, whatever, it shifts it to the left where they have more affinity for each other. This decreases oxygen unloading. It hoards the oxygen into the hemoglobin instead. So these two graphs kind of show that. Here's normal body temperature, or a body temperature graph. Normal body temperature is around 37, 38 degrees C. If I get hotter, then I'm going to have less affinity for oxygen and hemoglobin. If I get colder, it's going to shift to the left. They're going to have more affinity. We'll talk about why in just a second. Down here is carbon dioxide levels. If I have an increased carbon dioxide, oxygen and hemoglobin have less affinity for each other. If I decrease carbon dioxide, they have more affinity for each other. So that shifting makes a difference. And we'll talk about here why increases in temperature and carbon dioxide affect that. So as our cells metabolize glucose and use oxygen to make ATP, which again, that's a chapter that's coming up here soon, we're going to increase carbon dioxide levels because that is a byproduct of cellular respiration. Also going to increase acidity levels in our capillary blood. That declining pH is going to increase because the decline in pH and the increase in the partial pressure of carbon dioxide is going to weaken the bond between hemoglobin and oxygen. It's going to shift that curve to the right, and it's going to unload the oxygen where we need it most, which is where these levels of high carbon dioxide are. We need more oxygen there, right? That's called the Bohr effect. Same thing is true as metabolism increases, so does our heat production. It's one of the reasons why you get hot after you eat a big meal, because you're breaking bonds, you're releasing heat, and your temperature is increasing. And so as that happens, more metabolism, well, you need more oxygen as you have increases in metabolism because cellular respiration is happening and oxygen is a requirement for that. And so as temperature increases, which means higher metabolism, higher oxygen need, then that's going to decrease the affinity between oxygen and hemoglobin, and again, deliver oxygen where we need it to be at these high metabolically active tissues, right? Now, carbon dioxide is a little bit different story. It is transferred in the blood in 3 different forms. 7 to 10% is just dissolved in the plasma, as opposed to the 1.5% of oxygen. 20% is attached to the globin portion of hemoglobin. We call it carbaminohemoglobin. It's not attached to the heme, but it does attach to the hemoglobin. And then the rest of it, most of it, is transported as bicarbonate ion, or HCO3, in the plasma. Right now, how that happens, how we transport it as bicarbonate. Carbon dioxide combines with water to form carbonic acid, and that's H2CO3. This is just like any other acid, is going to quickly dissociate into H plus and HCO3 minus. Right? So there's an acid portion and a base portion. We call it the bicarbonate ion. So this is that reaction here. Carbon dioxide plus water, we get carbonic acid, and then we have the dissociation of this carbonic acid. This is reversible. All of this is reversible. We can make carbonic acid, dissociate it. We can bring those two back together and turn it back into carbon dioxide and water too. So both of those things happen. This occurs primarily in the red blood cells. where their enzyme carbonic anhydrase is located to quickly cause this reaction to take place. This can happen in the plasma, but it's really slow because this enzyme is not located there, right? Now in our capillaries, what happens is as we make this bicarbonate ion, okay, we've made carbonic acid, it's dissociated, so we have this HCO3 minus, it's going to diffuse out of the red blood cell into the plasma. But if I lose a negative charge from inside of a cell, if I just lose a whole bunch of negative charge and I don't replace it with something, that cell's going to have some issues. All right, so what we do is we bring in a chloride ion, which also has a negative charge. So you're spitting out a negative charge, but you're bringing in a negative charge. We call this the chloride shift. You're going to see this again when we get to the stomach, in how the stomach makes its hydrochloric acid. All right, so remember this for later when we get to the digestive system. So you have this outrush of HCO3. It's balanced as chloride comes into the red blood cell, and you can kind of see that here. I'm just going to focus you right here on this pathway where the bulk of our carbon dioxide is taken in. Brought into the red blood cell from the tissue, turned into carbonic acid when it combines with water. And again, this enzyme, carbonic anhydrase, is here to help this facilitate this reaction, make it faster. And then that acid dissociates into the bicarbonate ion in H+. So again, H+, it can join up with hemoglobin, but the bicarbonate ion leaves, it goes into the plasma, but then we bring in that negative charge of the chloride ion to offset that loss of negative charge. That's that chloride shift that we're talking about. And you'll also see up here, there's a little bit that's just dissolved in the plasma. There's a little bit that is turned into bicarbonate within the plasma, but it's slower because the enzyme is not there to help facilitate that. And then there's a little bit of carbon dioxide that is bound to hemoglobin itself. All right, so it's kind of showing all of those here. Now in our capillaries, our pulmonary capillaries, so this is where we're trying to get carbon dioxide out of our lungs and into the environment, right? So as bicarbonate ion comes back into the red blood cell, again, as we gain a negative charge, we have to lose a negative charge. So the opposite is happening here. We're bringing HCO3 minus into the red blood cell, and we're losing that chloride ion again. It binds to H+, forms carbonic acid again. The carbonic anhydrase enzyme splits it up into CO2 and H2O. And then that CO2 diffuses into the alveolus down this partial pressure gradient, and we breathe it out, right? So this is exactly the same thing that we saw a minute ago, except the arrows are pretty much all in reverse, right? We're bringing in oxygen because we're breathing in our lungs, but we're spitting out all this carbon dioxide that we made. So HCO3 comes into the red blood cell, chloride leaves, that forms or combines with H+ to form carbonic acid. Carbonic anhydrase splits it into water and carbon dioxide, and we spit that carbon dioxide out into alveolus. Right? The same is true up here with the other ways in which we make or carry carbon dioxide. All right, now the Haldane effect is talking about how much of the carbon dioxide is transported that it's affected by the partial pressure of oxygen. So when we have reduced hemoglobin, meaning it is not fully saturated with oxygen, it more readily forms carbaminohemoglobin. So the less oxygen a hemoglobin has, it more readily wants to combine with carbon dioxide and form carbaminohemoglobin. Right? So that just means that we can then carry more carbon dioxide in the blood. This encourages carbon dioxide exchange in our tissues and in our lungs because of this. So at the tissues, as more carbon dioxide enters the blood, then more oxygen dissociates from hemoglobin. That's the Bohr effect that we talked about earlier. More PCO2 causes a less affinity with oxygen and hemoglobin. Right? So, I'm talking about the sneeze. And no, I don't have corona. So, as oxyhemoglobin releases its oxygen, it more readily forms that bond with carbon dioxide to form carbaminohemoglobin. Right? So again, just encourages the exchange of these gases at the tissue level or at the lung level, wherever this respiration is taking place. All right, so how does carbon dioxide affect blood pH? We just talked about a minute ago how most of our carbon dioxide is transformed into carbonic acid, right? Carbonic acid is turned into an alkaline buffer that we call bicarbonate ion. It's called the carbonic acid bicarbonate buffer system. This helps to resist changes in blood pH. This is a big buffer system that I talked about in A&P 1 way back in the chemistry chapter, chapter 2, when we talked about buffers. So if you want to go back to look at chapter 2, you can look at that again. But this is a system where, remember, buffers are H+ sponges. They can soak up H+ if there's too much in the bloodstream so that the acidity is too low, and that'll bring the acidity back up. Or if it's too alkaline, it can squeeze out some of its hydrogen ions and release some of those and bring the acidity back down where it should be. So that's the job of a buffer. Our big buffer system in our blood is this bicarbonate. And so if H+ concentration is too high, the excess H+ is combined with HCO3 to make carbonic acid, right? And then as it begins to drop, then it dissociates and releases more of that H+ in the solution. And you think, back at this point, how in the world, if I'm making more carbonic acid, why is that decreasing, or why is that bringing pH back up? Because I'm making an acid, you would think it'd bring pH down. Remember that pH is only measured by free H+ ion. This H+, when it's put into this acid, is no longer free. It's bound to this thing, right? So it's not going to be calculated in the pH. So you're going to be able to bring that pH back up by doing that, right? Okay. Yeah. So HCO3 is an alkaline reserve of the carbonic acid bicarbonate buffer system. Right? So we can adjust our pH of our bloodstream really easily with this, really a waste product. Carbon dioxide is a waste product, but we're able to moderate and monitor our blood pH with it. Okay. Changes in respiratory rate and depth that affect blood pH. If you all of a sudden take really shallow, slow breathing, you're going to have an increase in the partial pressure of carbon dioxide in your system. That's going to drop your pH because you're going to make more carbonic acid. More carbonic acid is going to dissociate, making more free H+ ion, and your pH is going to drop. If you take rapid, deep breath, then that's going to decrease carbon dioxide in your system, and that's going to bring the pH to come back up, right? So we can change our ventilation based on what our blood pH is. If we need to adjust our pH, one of the ways we can do that is by changing our rate and depth of breath. Now, this is all automatically controlled, you know, autonomically, our respiratory center is taking care of this. Yes, we can have voluntary control of our breathing up to a point, but normally it's not something that we're thinking of, right? So depth is determined by how active the respiratory centers are. And then rate is determined by how long the inspiratory center is active. We can modify both of these, the respiratory center and the inspiratory center, depending on the needs of the body. And some of the most important things that affect this are the changing levels of carbon dioxide, oxygen, and acid, blood pH, really. And it's all sensed by chemoreceptors, central and peripheral chemoreceptors. Right? So for the chemical factors, the most important one is carbon dioxide. And you would think that oxygen would be the most important and potent stimulator of breathing. It's not. It's a buildup of carbon dioxide. Because remember, when we have even slight drops in the PO2 of our atmospheric air or whatever, our hemoglobin is still really saturated, right? But if you start to adjust carbon dioxide very little, it really starts to mess things up and really changes your ventilation. All right, so if blood PO2 or PCO2 levels rise, we call this hypercapnia. Then CO2 starts to accumulate in the brain. And just like anywhere, it turns it into carbonic acid, it releases more H+, the pH in our brain drops. That's sensed by central chemoreceptors in the brain stem, and then those synapses, the respiratory center in the medulla, and it increases the depth and rate of our breathing so that carbon dioxide levels go down, oxygen levels come back up, the pH comes back up, everything comes back to normal. So this is kind of the flow chart here. Arterial CO2 here. If you have increases in those, you'll increase the pH, you'll increase the acid, which is going to decrease the pH. That's sensed in the chemoreceptors. That sends the information to the medullary respiratory centers, that talk to the respiratory muscles, increases ventilation, and then everything kind of comes back to normal. You also have peripheral chemoreceptors that we'll talk about where those are located in just a second. They're also kicking in and doing the same thing. Now, partial pressure of oxygen, you do sense this in the peripheral chemoreceptors in the aortic and carotid bodies. So they're in the aorta and they're in the carotid arteries. they are O2 level sensors. When they're excited, they cause the respiratory centers to increase ventilation. So if you have a huge drop in O2, they're going to trigger the respiratory centers to increase. Declining PO2, again, though, doesn't have a big effect on ventilation because we have that huge reservoir that we don't use, basically, of oxygen to hemoglobin, right? When it leaves our tissues, we're still 75% saturated, right? So we got a big reserve there. But it requires a substantial drop in oxygen to see a change in ventilation. And we're talking about down to like 60 millimeters of mercury, which we don't see very much, at least in the outside atmosphere. Right? So here's where those peripheral chemoreceptors are. There's some in the carotid body here, and there's some in the aortic body, or the aortic arch. All right, the arterial pH. These can modify the respiratory rate and rhythm, even if carbon dioxide and oxygen levels are normal. These are mediated by those peripheral chemoreceptors in the carotid body, the aortic arch. And if you see a decrease in pH, that might reflect lactic acid buildup because of anaerobic respiration, maybe excessive ketone bodies, lots of things could happen. But the respiratory system has to compensate by increasing respiratory rate and breath and bringing that pH back up to normal. All right, so a summary of this. Carbon dioxide is by far the most important and most powerful respiratory stimulant. Any kind of increases in CO2 is going to increase rate and depth of breathing. Decreases of CO2 is going to be the opposite, right? Normal blood PO2 only affects it indirectly by chemoreceptor sensitivity. It influences the peripheral chemoreceptor sensitivity to changes in PCO2. But if PO2 falls below 60 millimeters of mercury, then it becomes a very powerful stimulant for respiration via those peripheral chemoreceptors. And changes in arterial pH, resulting in carbon dioxide retention or other metabolic factors, also act indirectly through those peripheral chemoreceptors. All right, so next I want to talk about pulmonary irritant reflexes. And so there's receptors in our bronchioles that respond to irritants. They communicate to the respiratory centers via some of the vagal, so the vagus nerve afferents, and they promote a reflexive constriction of our air passages. If you have a cough, right, very apropos that we're talking about coughs and lungs and all of that during this time, but the same irritant would cause a cough in the trachea or a sneeze in the nose, and they cause our bronchioles to constrict down. We also have an inflation reflex. It's called the Herring-Brewer reflex or inflation reflex. And this is when we breathe in, our lungs really inflate and stretch out. There are stretch receptors that are monitoring that stretch. And when they get to a certain point and they're stretched too far, they send an inhibitory signal to the medullary respiratory centers and they cause exhalation to occur. Right? Now, this is more of a protective response than it is a normal regulatory mechanism. These reflexes aren't in play in our normal, like, tidal in and out breathing. It's really just the, like if we just took a big deep breath in and held it, or we're just too much of a breath, like it's time to breathe out, then that's going to trigger and tell us to exhale. All right, so this is kind of a summary of all of that. We have stretch receptors in the lungs that inhibit the respiratory center. Irritant receptors also inhibit higher brain centers, voluntary control over breathing. Like I said, we do have control. We can start to breathe in really fast at will, or we can slow down our breath at will. Eventually, maybe to our own detriment, but at some point our brain is going to take over, cause us to pass out, and just do it. Other receptors like emotional stimuli, things like that, work through the hypothalamus to the respiratory center. And we have those chemoreceptors, peripheral and central, that are monitoring oxygen and carbon dioxide and acid. And we even have receptors in muscles and joints that talk to our respiratory center. So lots of factors are talking to our respiratory center and telling it, yes, breathe more or breathe less or whatever the case may be. So it's all working to keep us alive. And I think that is it. Yeah, that's all for this chapter that I want to cover. I'm going to post this. You'll see it. We'll have a discussion board. We have that review session going on for the exam. And again, you'll have seen the details on the exam by now at this point. So please let me know if you have any questions. I'm going to double check, see if this worked. And I guess I will see y'all then on the next one, the next the little lecture. I can't even think anymore. The lecture that I do. But again, I'll try to keep them as short as possible so you're not having to sit through a whole lot. You can come and go, watch these, stop them, pause them, whatever. Okay. It's kind of the benefit of online classes. I miss seeing y'all. I hope y'all are well. Wash your hands. All the things. Okay. Talk to y'all later.